study by T.J. Hammond shows that the levels of gene expression can change significantly in microgravity. The same study shows that cells differentiate much more in microgravity, and this allows scientists to make more detailed observations of the changes of levels in gene expression. Cancers are usually caused by the mutation of the p53 gene, a gene that prevents tumors, but a study by Venu Rahman shows that breast cancer is different because the HOXA5 gene encodes a protein that regulates the p53 gene expression. Loss of the HOXA5 gene expression is essentially what results in breast cancer tumors. A study by Blumenthal and Dennis shows that E. coli bacteria, the bacteria I want to use, levels of gene expression do change due to lack of amino acids in cultural conditions. My experiment tests two hypotheses. The first hypothesis is that the levels of gene expression in the two breast cancer genes will change in microgravity, and the second hypothesis is that the culture mediums do have an effect on cell differentiation and levels of gene expression. In the experiment, both of the gene plasmids are isolated and then co-transfected into the E. coli bacteria. I will use the group activation pack habitat. The control group is the culture without any culture medium, and then the 7 other tubes will contain an E. coli culture with 7 different amino acid solutions. The 3 separate fluids in each culture are a culture in stasis, a fluid that starts growth of the culture mixed with the amino acid solution, and the third is a fixative to stop the experiment. The level of gene expression will be determined from the mRNA found in the cells. This will show whether levels of gene expression of both breast cancer genes increase, and this is the level to which the rest of the cultures will be compared to. A study by Takeyo Onishi showed that the levels of gene expression for the p53 gene increases in space. Since the p53 gene is regulated by the HOXA5 gene in the case of breast cancer, I predict that the level of the HOXA5 gene expression will also increase. The experiment will allow scientists to study whether it is the structure of transcription factors or the way they bind to DNA that causes gene expression to change in microgravity and in different cultural conditions. The transcription factor that increases gene expression of the HOXA5 gene may allow scientists to find a way to increase p53 levels of gene expression without having too much p53 protein, which causes extremely fast aging that we want to avoid.